Welcome to the second half of the semester. What we are going to be doing is we are going to be taking for granted all of the concepts that we learned in the first half of the semester, and now we're going to be going into depth. We'll be doing more detailed case studies and really diving in and exploring some of these main concepts of evolution. Now remember, with evolution we have two main mechanisms: the mechanism of natural selection and the mechanism of genetic drift. So in this first unit, what we are going to do is we're going to separate those two mechanisms and really look at them in detail. I first want to start the second half of the semester by addressing a misconception. The misconception that exists is that evolution is always driving towards perfection. And when you think about this, there's a very good reason as to why so many people have this misconception that evolution is always changing in the lines of descent so that the newer organisms are more perfect, or the newer species are more perfect or more better adapted to the environment. And when we look at our first, some of our case studies during the first half of the semester, one of the case studies that we dealt with was during the epigenetics concept. We were looking at applications of that. We were looking at research showing epigenetic patterns of inheritance and the guinea pigs. Remember, the male guinea pigs were exposed to heat, and then that changed their gene expression so that their offspring were more likely to survive. Well, that sure looks a lot like driving towards perfection. When the context and the environment changes, the organism should make changes that then can be passed on to the next generation, because they're anticipating that the heat or that changed context is going to continue for their kids and potentially the grandkids and potentially the great-grandkids. This looks like driving towards perfection. So when we look at some of the other case studies that we covered, we've looked at how the stickleback fish, when the context changed, when the environment changed, they lost the spine. Remember, freshwater stickleback fish, if they still made that spine, that would have caused them to get caught by dragonfly larvae, less likely to pass on their genes. So any stickleback fish that would turn off the gene for making the spine would be more likely to survive. And then over many generations, we see that that's the only phenotype in freshwater ecosystems: stickleback fish that are missing the spine. We look at the ice fish. The ice fish is an extreme example where they took the gene, and through a repetition of that gene and the modification of lots of other systems, they were able to take advantage of a new niche. They were able to gather more resources, and that would make them more fit and more likely to pass on their genes to the next generation. So we've got lots of examples that we see across the planet where we do have organisms that are perfectly adapted to their environment. They seem to be completely in sync, or they make changes when the environment changes, and so it looks like there's this constant drive towards perfection. But this is definitely a misconception, because at the same time we're making observations of all of these organisms that can live in the extreme environments, can handle really incredible changes. At the same time we're observing those, we also observe within species and within populations within a species those deleterious alleles, those genes that cause disease or decrease an individual's fitness. We see that those persist, that those get passed on from one generation to the next. Now, when we look and we make that observation of, wait a minute, we've got a disease or an adaptation, or we've got some kind of symptom, we've got something that's based on genes, and that something is not increasing the organism's fitness. It looks like it's decreasing it. They're less likely to capture resources. They're more likely to become prey. Why is that persisting from one generation to the next? So some of the examples that should make us go, well, why does that exist? Will be things like really bright colors. So why do a lot of male birds have bright oranges, bright reds? They stand out in the landscape. When you've got predators around, why would you want to stand out? Like, that's not a good strategy. And yet that persists from one generation to the next. When we look at deer and moose and a lot of these kind of four-legged ungulate prey animals. The males, a lot of them are growing great big antlers. And when you think about the energy it takes to grow those antlers every year and then to carry them around, and not just to carry them around walking around in fields. No, they're going to go through forests with all kinds of brush and shrubs, and there's predators all around who are incredibly hungry. So if something is slowing you down, takes a lot of energy to make and a lot of energy to maintain, why would you keep doing this? This doesn't seem like a very good strategy. A lot of mating behaviors, running around, fighting with each other, attracting the attention of predators, potentially decreasing your own fitness because you're spending so much energy fighting and arguing and running around and growing new structures, and you don't necessarily have that energy for your own body. There are lots of stories out there at the end of breeding season of how many males die from sheer exhaustion of all of this battling and trying to defend territory. They weren't able to capture enough resources. So why would you go through it? Why would you do all of those behaviors just to pass on your genes? I mean, the males most of the time are going to take off anyways, so they don't care. They don't even know. They don't care about their kids. So what's the benefit here? What's the real, like, how is that driving towards perfection? Like, it doesn't make any sense. So a lot of adaptations we see are actually liabilities. And we have to take a deeper look at those. So in the lecture slides, I looked at some specific situations to humans where we see genes and we have these deleterious alleles that make an individual less fit. But the one thing you have to keep in mind when you're looking at those examples in the lecture slides is that the context changed. We had a population of humans that maybe was raised with wheat and a population that wasn't. And then the context changed for that population that wasn't raised with wheat. Now they're introduced to wheat, but they don't have the gene. They never developed the gene to be able to handle that. Same thing with dairy allergies. The hypothesis is that you have a group of individuals that was not around cows, did not spend a lot of time around cows. So why would they have the gene to break down the milk protein from cow milk when these humans only just recently started to get exposed to cow milk? Whereas the humans who did grow up with cows and did develop that relationship with them to use them not only for their meat, but also for their dairy product, which helped their survival, there would have been selective pressure. There would have been an advantage to developing the ability to break down the milk protein. But if you've never been exposed to it, why would that pressure have been there? Why would you have developed that in the first place? So we need to keep in mind always the context that the species lived in, and then within our species, we have multiple different populations. We have to see what all those populations have been experiencing, because populations are going to experience different abiotic and biotic factors, and that can contribute to whether or not we see the development of adaptations. Remember, adaptations are a lot of times rooted in genes and genetic mutations. So in this chapter, we address the mutation, that point mutation in the hemoglobin gene. So in the gene that makes hemoglobin protein, you can have the expected sequence of nucleotides that makes a hemoglobin protein in the appropriate shape. That shape then allows the red blood cells to be able to grab a hold of oxygen and bring that oxygen to other cells in the body and then get rid of the carbon dioxide. So it works wonderful. We get a point mutation, just a change in one letter, and all of a sudden, the protein has an unexpected shape. It causes the whole red blood cell to have this sickle shape. And then it can't carry oxygen and carbon dioxide efficiently. So the question is, why does this gene stick around in the human population? We have to go back to the context. If you live in an environment that has the malaria protist, the malaria protist wants to complete its life cycle in your liver, gets transported with the red blood cells, and if your red blood cells have that sickle shape, less likely for the protist to be able to complete its life cycle. In other words, if some of your red blood cells have that sickle shape, you are less likely to die from malaria. So we see that external forcing. The malaria protist in the environment is what forced or led to the change in our hemoglobin gene. Now, there was a drawback to that change. If both pieces of DNA in the homologous pair of chromosomes had that point mutation, most of the red blood cells are going to have the sickle shape. Well, then you're not getting enough oxygen to the rest of your body. So for that individual, the mutation sucks. But for the individual who has one piece of DNA with the point mutation, but the other piece of DNA does not have that point mutation, they get the best of both worlds. They get appropriately shaped red blood cells carrying oxygen and carbon dioxide, and then they get the sickle cell shaped red blood cells to help protect against the malaria protist. Yay for you! But what about the people who don't have the point mutation? Both pieces in that homologous pair of chromosomes have the expected gene, make the expected hemoglobin protein. Well, life is great for them as long as they don't go to tropical countries. Why? Because in tropical countries, there tend to be a lot of mosquitoes. And those mosquitoes are transporting the malaria protist from one organism to the next. And so if you happen to get bit and none of your cells have that sickle shape, you are more likely to contract malaria. That will decrease your fitness. So again, the context matters. If you have a homozygous genotype for the hemoglobin protein, the expected gene, no point mutation, then you got to live in the northern hemisphere. You got to live where there's winter. If you have a heterozygous genotype, just one piece of DNA has that point mutation, you're more likely to survive in the tropics. You have a high level of fitness, you're likely to pass on those genes to the next generation, but do be careful. If you are an individual who has a heterozygous genotype, and you mate with an individual who has a heterozygous genotype, the difficulty could be that 25%, or your children, your offspring, have a 25% chance of having a homozygous genotype. So if you're still in that tropical location, if your child gets that genotype, they could contract malaria and potentially no longer be with you. If your child gets both pieces of DNA with a point mutation, they will have sickle cell disease, and that's miserable for them. So there's a 25% chance of homozygous genotype, no sickle cell, no point mutation. 25% probability of both pieces of DNA having that point mutation and the child having sickle cell anemia. But there's a 50% probability that your offspring will have the heterozygous genotype. They get the best of both worlds. They get that malaria protection, but then they also have enough cells that are expected shape, with the expected hemoglobin protein shape. So, if you're in a tropical environment, this risk of potentially your child getting the homozygous genotype no point mutation, or homozygous genotype both pieces have the point mutation, sickle cell anemia, this risk is worth it because there is so much environmental pressure. This doesn't make any sense here in northern Illinois and in Canada and in places where there's snow, because our mosquitoes die in winter. And that helps stop the spread of malaria. Now, it doesn't mean that we can't have malaria here. As a matter of fact, through the Southwest, especially along the Gulf Coast there, where it's very, very wet and swampy, there used to be a lot of malaria. And this is where we get to look to the past and past practices where across our country they used to use a lot of DDT to kill the mosquitoes and to kill the insects. Now, maybe you've heard of DDT. Bit problematic there for birds. Once the birds had high concentrations of DDT in their system, that DDT would cause their eggshells to be very, very thin. And so the bald eagles and the hawks and the owls, when they would lay their eggs and incubate them, the mothers would squish and crush the eggs because the eggshells were so thin, And so we saw a huge decline. It took about 45 years for the red-tailed hawk, and especially the bald eagle population, to rebound here in our area, to the point where now seeing a bald eagle is getting to be kind of common. It used to be very, very rare. You never saw hawks or eagles in our area. So it took a while for the populations to recover from DDT, but the benefit of the DDT spraying was that we knocked back the mosquito population, and that then allowed us to be malaria-free. One of the considerations that we have to look at here in the United States is that our mosquitoes are starting to overwinter. They're overwintering in places like the subways of Chicago and the subways of Washington, D.C. These are places where they found some of the mosquitoes that we know transmit dengue fever, transmit malaria. And so it makes us a little nervous, because if our winters keep getting warmer, if we don't have those below zero cold snaps that start to kill some of our insect population, we may see some of these tropical diseases that we otherwise have been fortunate enough not to cope with. And then that brings us back to, if our context changes, could we anticipate that the frequency of the sickle cell point mutation, could we expect that the frequency of that gene might increase in our population here in Rockford or in Canada or North America? Could we expect for that to increase in our population? Well, it would be something we would start to watch over time. So do pay attention to the sickle cell case study, the video, and the Into the Jungle chapter. Make sure that you feel very comfortable with all of those details. There is a reason why we're going into that case study as such a wonderful example of so many different concepts. Not just the idea that what seemingly in one context looks like a deleterious gene, a gene that's decreasing fitness. In one context, it looks like it's a deleterious gene. In another context, all of a sudden you look at it and you go, wait a minute, maybe that's not a deleterious allele. Or maybe now I see and understand a little bit better why these genes get passed on from one generation to the next. So this brings me to the next set of concepts, which is artificial selection. Now with artificial selection, humans are selecting for specific traits. When we select for specific traits, sometimes what we are doing is we are enabling the fitness of an individual. So maybe there are deleterious genes or genes that decrease the individual's fitness, but we step in and we intervene. So our domesticated dog is a perfect example. Now, I love Shih Tzus. I think they are cute and adorable and wonderful. But if you've ever seen a Shih Tzu, they have that luxurious long hair and they're little small dogs. If you were to turn a Shih Tzu loose in the wild, the first thing that would happen is they would immediately be seen as prey. The hawks, the owls, the coyotes, even some of the foxes, all of them are going to look and view that little Shih Tzu as more of a bunny. They're not going to look at it and think, oh yeah, that's a wolf, I shouldn't touch it. No, they're going to think of it as prey. So if you turn one of those smaller dogs loose, the first, I think, most problematic thing is that they're seen as prey in the environment. The other problematic thing is that they've got all that long hair. They will pick up every single burr, every twig. It will get caught and tangled in their fur. And so if they aren't prey just by walking around, the fact that they get all bound up with burrs and can't move anymore means that they're less likely to capture resources. They might become prey. They might get infections and succumb to disease. We enable these little animals. We enable that long, luxurious hair. We enable them to be the size that they are. We enable their fitness. And so we have intervened, and all the bad genes come along with that intervention. Another beautiful example, I just saw a video a couple of weeks ago of a sheep in Australia. And with domestication of sheep, what we did was we selected for sheep that will continually grow their fur. What they should be doing is they should be shedding their fur every spring, getting rid of it. Makes sense. It's going to be a hot summer. Don't keep that fur around. But through artificial selection, we have been selecting for sheep that will continually grow their fur. We step in and intervene we become nature and shear them in the spring. And then it regrows and we shear them again. So we are stepping in and being their biology where their body would normally have done it before. So in this video, this domesticated sheep had gotten away from the farm and had been living out in the wild and had 70 pounds of wool. This poor thing was just loaded. You could barely even see its little face. It just had all of this huge matted wool around it and could barely even move. So with artificial selection, we step in and enable these organisms' fitness. Works out for us when it comes to sheep. I guess it works out for us with dogs, although sometimes it feels a little bit too high maintenance when you're dealing with something like a Shih Tzu or some of those more high maintenance little dogs. The other thing that we need to talk about is we need to talk about the types of natural selection. Now remember, with natural selection, we've got those criteria. Members of the population have variation. The variation is often based on genes. The individual will produce more offspring than the environment can handle. So the part of natural selection that sucks is that some of those individuals are going to die. And that over time, what we are going to see is that the majority of organisms in the population are showing similar genetics, showing similar adaptations, often show similar phenotypes. And that's the result of anybody who has an adaptation that doesn't help in the environment, that decreases the individual's fitness, should be eliminated from the population. But remember, they aren't always. So, we're looking at natural selection. We have three main types of natural selection. The first is directional selection. And with directional selection, what we see is that an extreme phenotype is being selected for. The environment most likely has changed. This extreme phenotype probably existed in the population, but was always relatively rare. You wouldn't see it very frequently. When the context of the environment changes, all of a sudden that extreme phenotype becomes more frequent. And if the change in the environment is dramatic enough, given a few generations, all you're going to see is that extreme phenotype. So we've got that directional shifting in our population towards our extreme phenotype. Our next type of natural selection is stabilizing selection. And with stabilizing selection, we see that the extreme phenotypes are detrimental. They're not going to be helpful or beneficial adaptations. Whatever those extreme phenotypes are, what we see is that they're getting eliminated from the population, and that most of the organisms are going to have similar phenotypes. They're going to have that kind of intermediate phenotype. And with stabilizing selection, if we're looking across a population of individuals and we think to ourselves, wow, they all look the same, we have to think in the back of our mind, well, maybe there's stabilizing selection here for that phenotype. And sometimes it's not just one phenotype, but it's multiple phenotypes. So maybe that's body size. Maybe that's coloration. Maybe that's behavior. Maybe that's food source preference. And we see stabilizing selection for a lot of different phenotypes. And from one generation to the next, we see everybody or every organism in that population being very, very, very similar. Our third type of natural selection is going to be diversifying or disruptive selection. And I like to think of this diversifying disruptive selection as being the opposite of stabilizing selection. Because what's happening with diversifying or disruptive selection is that we see that the extreme phenotypes are being selected for. That intermediate phenotype is not good anymore. It's being selected against. And so all of the individuals who have the intermediate phenotype are disappearing from the population, and we just have our extremes. And what can often happen with stabilizing selection, with diversifying disruptive selection, is that when we come across a population of individuals who are showing us this, and we've got these two extreme phenotypes, our knee-jerk reaction is to say, these are two separate species. When we get a hold of the DNA and we start to make some more detailed observations, we often realize that no, they're the same species. It's just two extreme phenotypes that are being selected for. And this is why we're jumping in to the rock pocket mouse case study, and why you want to make sure to look at the Oldfield mouse description in the textbook chapter as well, because we've got two beautiful examples where the knee-jerk reaction could be to say that wow, we've got different species of mice, and yet when we look at this, what we see is that based on the context, based on the environment and the role of the predators, the extreme phenotypes are being selected for, and anything in the middle is going to go away. Now, if you have any questions on these types of natural selection, if you have any questions about the Hardy-Weinberg model, you've got the video and you've got me for help. Please, please, please reach out for any problems that you might be having. Stop into the Zoom office hours. I want to make sure I can help you with these really more detailed concepts. We can no longer be vague. We have to get into the details.